Hi everyone, thank you very much for joining this live Q&A. Today I have with me Caroline Archer, Pedro Chianto Pinto, and Stephanie Corella. Stephanie, I'll begin with a question for you, if you don't mind. So our first question is, have permeability studies also been performed with colonic organoids?
Yeah, good question. Not in this study, no. So not with those permeability markers we have been using there. We had some other studies where we were basically looking as a first step into donor variability into the colon. But only on a high level, I can say we were looking at 6 donors and there's quite some variability we use one specific high permeable compound or medium permeable compound. And what we saw there was a large donor variability between those 6 donors. But a matching data set for the colon, for the data I presented, we don't have at the moment.
Thank you very much. Pedro, what are the main factors that implicate drug absorption in your models? Oh, sorry, you're muted.
Oh, sorry about that. Yeah, so there are several ones and some that we probably haven't anticipated, but one is definitely the chips themselves and the material they're made of. So the ones we're using are made of PDMS, so they work quite well for low permeability drugs or low binding drugs, but especially drugs that are protein bound, there might become an issue because some of the drug can be trapped in the system, and that may kind of skew your calculations if you want to determine how much drug actually gets to the cells or to the organoids that you have inside the system. So yeah, so that would be the main issue, and the other one would be to understand how the chips are perfused, considering your perfusion rates or your cycles of perfusion, and how that affects how much drug is delivered to the structures themselves. Yeah, so that requires so one way we did in the past or recently actually was to model how the flow in the system can work, and the other one is could be just to let your system saturate, so run it for a couple of hours and then assume you're at a saturated state when you run your experiments. Yeah.
Thank you very much. Caroline, question for you. What luminescent approaches you use to measure cell viability? This question, they say that they use LDH before, but it only can provide with cytotoxic rate.
Yeah, so we use ATP luminescent assay, so the cell titer glow from Promega for our microtissues. So because we're in the state at the moment of just developing a cardiac MPS model, we haven't set our sights on anything, but we would be looking for something that is sort of be able to take continuous samples on rather than destructive, which is what the ATP assay is. So but the beauty of cardiac tissues is that you get spontaneous beating, so it's very clear to see if you've got any sort of immediate toxic effects happening. So yeah, we've used ATP, but we were looking to use a multiple range of markers, so the functional beat rates, LDH leakage, and also other biomarkers which leak from cardiac cells, so troponin, for example. So we'd use a multiple range to get a broader view of what's happening.
Thank you very much. Etienne, question from you. Have any data with the organoids been produced under hypoxic conditions?
No, not yet. It's something we're looking into. Let's say the installation for hypoxic conditions are quite, well, it needs a full installation, and we don't have either the space or the let's say the proper installation to set it up.
Thank you very much. And Stephanie, back to you. How do you measure drug metabolizing enzyme activities, and how does this compare to protein expression data along the intestine?
Yeah, so we have measured basically metabolite formation in our samples with the organoids just by mass spectrometry. We have not performed the abundance measurement, so proteomics in-house. So there we can just compare to data available specifically from the Oswald group. For example, they did a lot of abundance studies for the drug metabolizing enzymes, also transporters for colon and small intestine. So that's kind of where we benchmark ourselves towards to at the moment.
Oh, thank you. Pedro, what plans do you have for your future models? What would you say?
Well, other than a continuous development and trying to sort out all the issues that we came across every time we try something new, so implementing or trying to source human tissue to grow, especially cancer-derived tissues, will be the next step. So we need to implement a workflow for this. Since we're based in a hospital, we can access surgical material. So the idea is to use this excess material that is only to diagnostics or pathology, and we actually don't need much because the system we're using, the perfusion chambers are about the size of a 96 well. So we wouldn't need much, and we could access. So surgeries are done weekly, so we could have a decent pool of patients. It's just a matter of implementing how we can do this in practice, from collecting the samples to bringing them to the lab to processing them, and also to understand how long we could grow the samples. So we're mostly interested in secretion out of cancers. So it is interesting for us to see how long we could fuse them and how long we could collect these samples. But at this stage, we're still optimizing a lot of this just using standard cell lines and spheroids that we can grow and so on. So.
Amazing, thank you, Caroline. What approaches are used to access cell health of 3D and MPS models?
Yeah, so I guess similarly to what I answered before, was looking at either sort of ATP content, which is a good measurement of cell health, or LDH, which is sort of more cell toxicity, using those, but also leakage of the structural proteins like troponins. So, and also more morphological assessments as well. So using sort of immunofluorescence approaches to look at general cell health that way.
Thank you. And then Etienne, what would you say are your key accomplishments over the years using 3D cell culture models?
Oh, key accomplishments. The fact of establishing, I would say, co-cultures to mimic more closely the tumor microenvironment is quite an important stepping stone, because it does improve in the way that we think or develop all our cell therapies and living therapies. So I think this is quite important, and we're looking into other ways to make it even more close to the tumor microenvironment and mimicking more closely the patient context. Other important so the fact of being able to do something that I actually quite passionate about is studying the metabolism of organoids and tissue samples, and being able to access this kind of technologies and apply and develop the pipelines to analyze all those samples on the CyTOF and the MSI to actually get, I think, very important information on the tumor microenvironment.
Thank you. Stephanie, in your transporter assay, which one is most predictable, CaCO2 versus the organoid-based system?
Sorry, can you repeat the question?
Yeah, of course. In your transporter assay,
yep,
which one is more predictable, your CaCO2 versus the organoid-based system?
The CaCO, okay. The
yeah, sorry,
CaCO2 versus.
Sorry,
I didn't understand. Yeah, of course, on the CaCO, there is already I mean, there's a huge data set available, historical data in-house, historical data available from other labs. So obviously that is more predictable. Also there we have, let's say, characterized even in-house 30, 40, 50 compounds with historical data, where we then also get a good dynamic range in which this CACO2 performs. And there for the organoids, this is basically all not established. So we used here really just a minimal compound set. And there it looks, if we just look at high and low permeability marker, that the dynamic range is a bit smaller than for CACO2, but that can depend on many aspects which we are still looking into. So is it the time of the differentiation frame you give to the organoids? Is it the donor? Is there again also a donor variability? And you will get different dynamic ranges for different donors. This is all something we haven't explored, and we would need there a much larger data set for the CACOs for the organoids to really compare. So of course, at the moment, the CACO2 is our gold standard, and those data, we understand them well.
Thank you very much. Caroline, I've also got another question for you. Is it possible to run a kinetic study, such as activation of signaling pathway using cardiac microtissues?
Yeah, that's something which would be amazing to be able to develop. So I briefly mentioned in the talk what Fei Gao, who is our postdoc student in our team, what he's up to. So he's using IMC to try and do this, but I guess the main drawback of that is that you've got fixed. So you need to take sort of multiple endpoints in order to try and get that kind of kinetic transient understanding of what's going on cell signaling wise. So you can look at like phosphorylation events of various kinases that you're interested in. And there have been some that are implicated in structural cardiotox. So yeah, if there's, we don't have a way yet, but that's sort of getting us more closely there and enabling us to look at a more single cell resolution, so we can look at the 3 different cell types in our cardiac microtissue. So that's something that he's working on currently.
Amazing, thank you. And then my final question is for Etienne. And what are your plans to further evaluate the translational value of your lung organoids?
Very good question. So we're kind of building a full set of characterization of omics around the organoids and from the samples that have been derived from. And because let's say that organoid is quite a very recent field, and there's still a lot of things that we are not fully aware of what our organoids are able to do, what they are able to mimic, in which assays that they might be the most relevant model to use, and some other assays where it's maybe too complex, too heterogeneous to give you a black and white answer. So there's plenty of different transcriptomics, proteomics, secretome, where we are trying to understand as much as we can everything that is surrounding the organoid model, and try to understand that. And afterwards, try to see how much of our organoids we're able to, for example, transplant into in mice for PDX assays. And we would have then the in vitro and the in vivo, let's say, platform where we would have a direct translation between both and see and try to understand the different correlations between the two.
Thank you very much. Unless you've got any questions for each other, that will be the end of the Q&A. Any other questions at all? Perfect. Thank you very much for joining.